Hello and welcome back. This lecture we will cover the rest of Chapter 10, from DNA to protein, gene expression. Last lecture we focused on transcription, the process of producing RNA from DNA. In this lecture we will cover translation, which is taking the code from RNA produced in transcription to produce proteins. There's a video of translation posted on Canvas that I recommend you watch after this lecture to help you better visualize the process. So what are we waiting for? Let's get started. Translation is the process of translating the nucleotide sequence of an mRNA into an amino acid sequence, which occurs in ribosomes. In prokaryotes, transcription and translation are coupled, the ribosome binding to the messenger RNA as it's being transcribed. In eukaryotes, the nuclear envelope separates the mRNA production and transcription from translation. The genetic information is read in a series of 3 nucleotide bases called codons. Each codon specifies an amino acid. AUG codes for the amino acid methionine and is called the start codon. The mRNA codons UAA, UAG, and UGA are called stop codons and end translation. For most amino acids, there's more than one codon. Genetic code is referred to as redundant because of this. This is table 10.11 and is the key for decoding each mRNA codon. You'll be able to use this table on the final exam. The way it is read is quite simple. If you have the codon ACU, you find the first letter A on the left side. Then you look for the second letter C on the top, and on the right is for your third letter U. So to find ACU, find it here in the middle, and that codes for the amino acid threonine. The genetic code is said to be nearly universal. The codons that specify amino acids are the same in most organisms, with exceptions that occur in mitochondria, chloroplasts, some protists. The genetic code is fairly old in that it unifies life and indicates that all life came from a common ancestor. Point mutations also confirm the genetic code. Point mutations is the altering of one nucleotide, as we saw a couple chapters ago, a coding region and compared with the amino acid sequence and the resulting polypeptide. Silent mutations can occur because of the redundancy of the genetic code mentioned earlier. For example, if CCG is changed to CCU, both codons translate for proline. On this slide you have the DNA molecule on the top, the transcribed messenger RNA molecule in the middle. This RNA molecule produces the polypeptide chain on the bottom. This is the same DNA molecule from the last slide. However, the highlighted nucleotide is changed from a C to an A. The resulting mRNA molecule still codes for the same polypeptide chain, because this is a silent mutation. However, not all mutations are like this. A missense mutation results in a change in the amino acid sequence. GAU is translated into aspartic acid, but if it's changed to GUU, the amino acid is translated to valine. Here's the same DNA sequence from before, but the resulting point mutation changes the GAU to GUU, producing valine, which is the change of the amino acid sequence, making this a missense mutation. A nonsense mutation results in a premature stop codon, which results in a shortened protein that is usually not functional. Here's our same DNA molecule from before, but a codon is changed to a stop codon, resulting in a small polypeptide chain, thus making this a nonsense mutation. Frame shift mutations are insertions or deletions of one or more bases. This will cause a new set of triplets to be read and result in an altered sequence of amino acids and resulting polypeptide. Here's our original DNA molecule again, with an extra thymine added in. This new addition completely changes the amino acid sequence beyond the point of insertion. Okay, here's our first progress check. You'll need to use figure 10.11, the genetic code, to figure the first question out. Practice using it, because there will be an assignment and the final exam will require you to use it as well. Take your time to answer these questions and move on with the lecture when ready. Okay, we covered the genetic code, mutations to the code, and how it affects the codons. But how does translation actually happen? Well, it cannot happen without the help of transfer RNAs, or tRNAs for short. tRNAs link information in mRNA codons with specific amino acids. tRNA delivers the correct amino acid that corresponds to each codon. For each codon, there's at least one specific type of tRNA. The structure of tRNA helps with its three functions. It binds to a specific amino acid and becomes charged. tRNA will bind to the mRNA via the tRNA's region called the anticodon. The anticodon is a nucleotide sequence that is complementary to the mRNA codon for the particular amino acid. Also, tRNAs interact with ribosomes non-covalently, which is important in their reuse for future reactions. Here's the structure of tRNA. The amino acid binds to the top of the tRNA at the amino acid site. The anticodon is the bottom of the tRNA, is complementary to the mRNA's codon. tRNAs have what is called a wobble effect, or just wobble for short. This means that the specificity for the base and the 3' end of the mRNA is not always observed. In other words, codons for alanine, such as GCA, GCC, and GCU, are recognized by the same tRNA. The codon's first two letters, G and C, are the same for alanine, but the final nucleotide in the codon is recognized by the same tRNA. This wobble effect allows cells to produce fewer tRNA molecules, which can save on energy and time to produce protein. tRNA becomes charged by the enzyme amino acetyl tRNA synthetase. The amino acid enters the active site, while the tRNA molecule enters the enzyme as well. The enzyme pairs the amino acid with the right tRNA molecule, charging it, and making it ready for translation. Translation then occurs at ribosomes. Ribosomes consist of one large and one small subunit, and exist separately when not translating. Ribosomes are not specific. They can make any type of protein. Figure 10.14 shows the structure of a ribosome. The small subunit is on top with the large subunit on the bottom. There is an mRNA molecule going through it for translation. The large subunit has 3 tRNA binding sites that go in the following order: the amino acid site, A, polypeptide site, P, the exit site, E. These are usually abbreviated as APE. The A site binds with the anticodon of a charged tRNA molecule. P site is where tRNA adds its amino acid to the polypeptide chain, and the E site is where the tRNA sits before being released by the ribosome. Translation occurs in 3 steps: initiation, elongation, and termination. We will go over these steps in the next few slides. Initiation is when the initiation complex is formed. This is when the small ribosomal subunit binds to an mRNA recognition sequence. Methionine charged tRNA molecule then binds to the AUG start codon. Remember, as we mentioned earlier, methionine is the first amino acid because AUG is a start codon. The large ribosome subunit then joins the complex with the charged tRNA in the P site. The complex is then put together by a group of proteins. Here is a picture of the eukaryotic cell. Remember, transcription occurs within the nucleus. And translation occurs in the cytoplasm, where the ribosomes are. During initiation, translation, the mRNA binds with a small ribosomal subunit as seen on the top. Methionine-charged tRNA binds to the mRNA and small subunit. The large subunit binds to the small subunit with the methionine in the P site. After initiation, next step in translation is elongation. Another charged tRNA enters the A site, and a large subunit catalyzes 2 reactions. First, the bond between the tRNA and its amino acid in the P site is broken. Next, a peptide bond forms between the amino acid in the P site with the amino acid on the tRNA in the A site. When the first tRNA releases this methionine, the ribosome then moves so that the tRNA in the P site moves to the E site. tRNA will leave the ribosome via the E site, so it may be charged again. Elongation occurs as the steps are repeated. Figure 10.16 shows elongation. First tRNA always has methionine and starts in the P site. As a new charged tRNA enters at the A site, a peptide bond is formed between the P site tRNA and the A site tRNA. As the ribosome moves along the messenger RNA, the first tRNA without its amino acid moves to the E site and is released from the ribosome, so that it can be used again. The process occurs again as the tRNA at the A site is now at the P site. A new tRNA enters at the A site, forming a peptide bond between the amino acids. This process will keep occurring to grow the polypeptide chain. Termination of translation occurs when a stop codon enters the A site. It binds a protein release factor, which allows hydrolysis of the bonds between the polypeptide chain, the tRNA, and the P site. The newly completed polypeptide then separates from the ribosome. Figure 10.17 illustrates termination of translation. A stop codon enters the A site, and a release factor will enter the A site as well. When the release factor enters and binds to the ribosome and mRNA, it breaks the bond between the polypeptide tRNA, ending translation of mRNA. Eventually, the subunits of the ribosome dissociate and break apart with the mRNA, ending translation. Table 10.2 is a good review of some of the differences between signals that start and stop transcription and translation. Be sure to understand these two processes. Sometimes students will confuse translation and transcription. I recommend reviewing your notes and watching the videos on Canvas to help you study this material. The example of translation that we looked at showed only one ribosome translating a messenger RNA molecule. In reality, several ribosomes can translate a single mRNA molecule. This is one way that cells can produce a protein more quickly. Here, figure 10.18 is showing multiple ribosomes producing polypeptides. You can see that researchers took an electron micrograph of this as well. In it, you can see the dark circles, which represent the ribosomes translating messenger RNA and producing polypeptide chains coming off of them. Here's another progress check. It's kind of a vague question, but I want you to understand the steps of translation, which are initiation, elongation, and termination, and what happens in each one. Take your time to answer it and move on with the lecture when ready. After translation, the protein is usually modified. It may be modified during and after translation. As it emerges from the ribosome, it may fold into its 3D shape and start performing its functions right away. Some polypeptides may contain signal sequences to target them to the nucleus, mitochondria, plastids, and peroxisomes. Figure 10.19 illustrates all the places polypeptides may go in the cell. Signal sequences will direct it where to go. If the polypeptide is directed to go to the rough ER, translation will pause and the ribosome will bind to the rough ER membrane. Translation will resume with elongation continuing. Polypeptide will cross into the rough ER's lumen. Polypeptide may stay in the ER or be sent to other regions within the endomembrane system. Most polypeptides are modified after translation. Proteolysis is the cutting off of signal sequences, or cutting a long chain into smaller polyprotein chains. Glycosylation is the addition of carbohydrates to form glycoproteins. Phosphorylation is the addition of phosphate groups by protein kinases. Charged phosphate groups change the structure of the protein to expose binding sites. Figure 10.21 is illustrating several ways the polypeptide chain may be modified after translation, such as in proteolysis, glycosylation, and phosphorylation. Okay, last progress check for the chapter. Take your time to answer this question and move on to the last few slides. If you remember from last lecture, we had the opening question, how do antibiotics target bacterial protein synthesis? Some antibiotics, such as tetracycline, bind to small subunits of bacterial ribosomes. They will bind to the A site within ribosomes. You can see tetracycline nudged into the 3D structure of this ribosome. Here, tetracycline is the little blue dot in the middle. By binding to the ribosome, tetracycline will stop protein synthesis. However, strains of MRSA that are resistant to tetracycline are emerging. As we saw earlier with bacterial conjugation, bacteria can pass antibiotic-resistant genes on plasmids between them. These resistant genes may encode for proteins to transfer tetracycline out of the cell, or genes to prevent tetracycline from binding to the ribosome. To overcome this resistance, new antibiotics are being developed and tested. In a sense, there is an arms race between drug resistance and antibiotics that will always continue. Well, with that out of the way, that concludes Chapter 10. Please, as mentioned several times before, review the videos of transcription and translation on Canvas, as I think they will help you. Please let me know if you have any questions on the material. Thank you for listening, and have a good rest of your day.